So hi, everyone. Thank you so much for joining us for our first ever ECR Careers event on potential careers that are available after doing a PhD or a postdoc. So Proteintech is really excited to be hosting this event in collaboration with Active Motif, who are leaders in providing life science tools to study epigenetics. So today we have 5 amazing speakers here who will share their unique journeys that led them to the careers that they currently have today. Before we pass it off to our speakers, though, I just want to share a few company slides so that you know who we are as Proteintech. So Proteintech is a leading manufacturer of antibodies, proteins, and immunoassays across several research areas. The portfolio comprises of 3 core product lines. So we have our Proteintech branded antibodies and immunoassays. So we have a very expansive portfolio of antibodies. We also offer antibody labeling kits, ELISA kits, and IHC kits. We also have our humankind branded bioactive proteins, and all our proteins are expressed in human HEC293 cells so that you can be confident that they have native folding, post-translational modifications, and glycosylation. And then finally, we have our Chromatech branded reagents that are based off of on alpaca nanobodies. And our nanobody-based reagents are ideal for immunoprecipitation, immunofluorescence, or protein purification. So the product lines all share common values under the main Proteintech brand. So we have high quality and validated products to ensure scientific excellence. But each portfolio comprises and addresses its customers in a separate, specialized manner. I also just want to take a moment to celebrate the success of our customers because our customers' success is also our success. And we're really proud that Proteintech products have been cited in over 180,000 publications worldwide. We're also really proud that we were the winners of the Researcher's Choice Award last year from CiteAb. What makes Proteintech different from other companies is that we manufacture all our products in-house and sell directly to our customers. We're also very transparent about our product performance and include original validation data. So with that, I just want to share the outline for today's ECR event. So we will begin with our keynote speaker, Dr. Lisa Bouchier-Hayes, who is an associate professor at Baylor College of Medicine. Afterwards, we'll hear from Dr. James Quinn on what it is like to be a translational scientist too, and he's from Eisai. Then we'll move on to Dr. Harry Potter, who is a teaching lecturer at University of Central Lancashire. And then Dr. Vrutant Shah, who's a research associate too at Methodist Research Institute. And finally, we'll finish with Dr. Rwik Sen, who's a field application scientist at Active Motif. So after all the talks, we will do a panel Q&A with all the speakers. So feel free to input any questions that you have in the Q&A box as the speakers are giving their talks. So with that, it's my pleasure to introduce our first speaker, Dr. Lisa Bouchier-Hayes. So Dr. Lisa Bouchier-Hayes is an associate professor in the Hematology Oncology Division of the Department of Pediatrics at Baylor College of Medicine. She obtained her PhD in molecular cell biology from Trinity College, Dublin in Ireland. She moved to the US in 2002 to do a postdoc at La Jolla Institute in San Diego, followed by a second postdoc at St. Jude Children's Hospital. Her research focuses on investigating the molecular mechanisms of cell death, specifically mechanisms of caspase activation and non-apoptotic functions. Outside of her lab, she is passionate about faculty development, helping the junior faculty in the department to navigate the NIH grant system. So with that, I will go ahead and pass it off to Dr. Bouchier-Hayes. Thank you so much for being here today.
Thank you very much. Okay. Oops. Can you see that okay? Okay. Yes. Thank you for having me here and giving me the opportunity to talk about my journey through on the academic research track. So I wanted to start here. When I was a PhD student, I went to a career symposium very much like this, and a lot of the speakers presented this kind of graphic where it shows a very linear way of getting through academia, where you graduate from university, you go to grad school, you do a postdoc, you become a PI, and then a subset of them go on to become superstars that are really well known in their field. So as I said, this is like a fairly linear way of thinking about things. And for every person that goes through this kind of process, it's always very different. And it tends to be more like this, where there's a lot of bumps and corners along the way. And that has kind of been the story for me. So as I tell my story, keep in mind that, you know, a lot of the decisions I made weren't so much strategic as just haphazard. But I hope to give you some advice of what I learned as I went through this. So I started in Trinity College, Dublin, doing my undergrad degree. I'm from Ireland. And I did a degree in genetics, and part of the genetics program, they had this summer internship scholarship program where they would send us to the States to work in a lab. And I was sent to Shearing Plough in New Jersey. This is a pharmaceutical company that I think no longer exists, but they make Claritin, and I was working in the quality control microbiology department. It wasn't the most exciting work, but it did make me interested in doing a PhD because I liked the lab work, I liked the environment, and at that time I wanted to go into industry. Another thing that inspired me was my father. He was a surgeon in the College of Surgeons in Ireland, and he, unusual for the time, he was one of the few surgeons who kind of started doing research in like cell-based models and stuff. And he told me about this newfangled thing called apoptosis. At the time, it wasn't very well known. And he was like, this is something really cool that you should look into. In my final year of college, we had to do a literature review as part of our final year assignments. And one of the options was on the genetics of apoptosis. So I took my dad's advice and I decided to look more into it and wrote the review. And I got really interested in this process of cell death. So then all I knew was that I wanted to do a PhD on apoptosis. There were two people in the country at the time who had labs focused on apoptosis research. So I applied to both of them. I got accepted to one of them. And shortly after I started my PhD, I became less interested in going into industry and felt I was more suited to a role in academia. So I did my PhD also in the genetics department in Dublin, and focusing primarily on apoptosis, which is a form of programmed cell death. And this tree is shown here because the word is from the Greek, describing the falling of leaves off of a tree. It is a form of cell death that allows for the removal of damaged extra or unwanted cells from the body without engaging the immune system. The lab I was in was heavily involved in gene discovery because at the time, most of the proteins that were required for regulation of these pathways were not known. And we discovered a protein that we called cardinal. And in the first unexpected turn to my research, we found it wasn't involved in apoptosis at all, but it was an inhibitor of a different pathway called the NF-kappa B pathway. So when I say we discovered it, we weren't the only ones. It was published by two other groups, one before us and one after us, all in the span of about 6 months. Each paper had slightly different conclusions, sometimes vastly different conclusions, and they were all published in the same journal. So I'm just putting this up because the idea of getting scooped is a big fear among many of us, and I have gone through it, and it's not always the worst thing in the world, and you usually can still publish your stuff, and sometimes even in the same place. So as I was doing my PhD, I became interested in moving to the US, and there were a few reasons why. Firstly, I wanted to continue working on the cell death field, and the leaders in the field were in US labs. And I also wanted to experience working in a new country and the science in the US, and having worked over a few summers in during my undergrad, I was I was interested in going back there. And so for applying, what I did was I made a list of investigators that I wanted to work with, and I applied to one of them, and that was the mentor of my PhD supervisor. And I was offered a position. So at the time, the advice that I got from my side of the ocean was, this is the best offer you'll get. Don't negotiate for travel money or salary or anything. You'll put them off. And they won't pay for you to go to interview because it's a transatlantic flight. This clearly was terrible advice. What I've learned and what I tell people now is good postdocs are really hard to come by. If you're interested in doing a postdoc, you will not have a problem finding one. You can always negotiate and ask for what you want. It's not going to cause anyone to pull an offer or be less interested in you. Apply to more than one place, even if you have a favorite place that you want to go to, because it will give you an idea of how competitive you are for these kind of positions and what you can ask for. And most importantly, never go somewhere sight unseen. If you are coming from Europe and you want to go to the US, it might be difficult to get them to pay for you to go there. But if you can go to a meeting that's in an adjacent city or something, they will the lab you're applying to will be more likely to fund you to go on a shorter trip. So there are different ways of going about it, but you should always check out the place if you're considering a move. I moved to San Diego, the La Jolla Institute, to start my postdoctoral fellowship. The lab that I moved to was very focused on, they were pioneers in interrogating and uncovering details of the mitochondrial pathway of apoptosis. And this is where release of cytochrome C is essential for many apoptotic pathways leading to cell death. And they were also had done a lot of early work in using confocal microscopy and time-lapse microscopy to analyze these pathways so we could see what was happening in real time. So we use proteins like GFP that are isolated from jellyfish. And this is the structure of GFP. It has a really nice barrel structure that cages the floor in yellow in the center of it. And because we were in San Diego, I like to show this San Diego beach scene, which was made by Roger Chen's lab. And it is bacteria expressing different colored fluorescent proteins all streaked out in a Petri dish. And their lab was, in an institute around the corner from us. So I got to see him talk one time. They did some amazing stuff. And he went on to win the Nobel Prize for the discovery of GFP. So we can use these tools to look at apoptosis and look over time at cells dying. So you can see green is cytochrome C, it's getting released, the cell shrinks, and lots of other colorful things happen, showing different events that occur during apoptosis. So my specific project was to microinject liposomes into cells and see if they release their contents during apoptosis. And it was just as crazy as it sounds. So the idea behind it was that we had shown, at least other people in the lab had shown, that lipid vesicles can mimic mitochondria in vitro, and we wanted to see if we could recapitulate that in cells to investigate the requirement for membrane proteins in cytochrome C release from mitochondria. Long story short, the project never worked, at least not well enough to publish. So it was a lot of work that really never went anywhere. But at the same time, I learned a lot of new techniques, and through collaboration with other lab members, I was able to be co-author on a number of really good papers. And that was because I was the only person in the lab who could do microinjection, and I was one of the few people in the lab who could do confocal imaging. So it could put me in a good spot to get these things working. So then in around 2005, my boss was recruited to St. Jude Children's Research Hospital. And I wasn't that interested in moving to Memphis from San Diego, but once we saw the place, it was hard to turn down because St. Jude is a really amazing place to work. After a little bit of struggling still with this liposome project, I switched projects to focus on the caspases that are involved in apoptosis and generate new tools to measure activation. So what we did is we took a fluorescent protein like I showed before, and this one is venous, and it can be split into two fragments that are on their own and not fluorescent. And we fused each fragment to a part of the caspase. And these caspases get activated by dimerization when they get recruited to large molecular weight protein complexes. So when they get recruited, the two halves of the venous come together, refold, and light up. So we can look at this really nicely over time. And the red is the mitochondria here, and the green is the venous coming on, representing caspase activation. So finally, I had a project that was working, and we published it in a molecular cell in 2009, September 25th. I still remember the date it came out, and it made the cover, which was really awesome. So it was around that time that I started thinking and applying for faculty jobs. Now, if you're following along, I've been 7 years at this stage in a postdoc, and some of you might be wondering, why did I take so long? Well, first of all, it was very practical. I had no first author paper, but also I felt like I wasn't cut out to be a PI, whatever that means. And I think this is something that a lot of us, especially women, tell ourselves. And to my mentor's credit, when I said this to him, the first thing he said was, why not? I think you would be. So it was a bit of a slow burn, but I started, like, unconscious to myself, imagining how I would do things if I was in charge, how I would set up a lab, how I would teach people how to do stuff, what kind of projects I would work on, and slowly realizing that, like, I was, quote unquote, cut out for it. So initially our plan was to return to Ireland. I wrote, unfortunately, this was around the time the crash happened, so the country went bankrupt and there was a national hiring freeze on. I wrote several grants to try and circumvent this, so if I could pay my own way, they would create, the universities that I was applying to would create a job for me. Most of them were not successful. And I was eventually offered a lectureship. At the same time, my husband's boss was recruited to Baylor College of Medicine. My husband is also a scientist. So he negotiated faculty positions for both of us. So at this stage, we had a choice to make: Ireland, moving back home to Ireland versus Texas. So I just want to show the comparison of the two positions and what we were being offered. The Irish position, there really wasn't any startup. They would match 50,000 euros if I got a grant for a PhD student. The US position was a generous startup for 3 years, including equipment and my own confocal microscope. No staff positions versus a postdoc and a technician. No information about space or what space I would get, versus being told I would get 750 square feet of lab. A shared office versus my own. Heavy teaching load versus none. And importantly, no help to get my spouse employed versus positions for both of us guaranteed. But even so, there was the idea of being able to move back home and closer to our families versus moving to a new city in the US and starting over again. But once we saw Baylor and everything it had to offer, it really was a no-brainer. So we decided to move to Texas and Houston. So I established my lab in 2011 and got my name on the door, and I was very excited about this. And this is my lab now. As it stands, it's a really talented group of individuals, and I feel very lucky to be able to work with them every day. So just to sum up the last almost 12 years, We've trained 5 postdocs, 4 graduate students, 6 technicians, 12 rotation students, 1 visiting graduate student, 3 med students, 7 undergrads, 1 prep student. So that's the post-bac course that they have here at Baylor. We published 24 peer-reviewed papers, 5 book chapters, 2 online video protocols, 2 webinars, including this one. We've seen 3 weddings, 4 babies, and 1 puppy. So the puppy belongs to my graduate student, Carla, and she named her caspase, and I very quickly made her our lab mascot. And importantly, we've raised over $2 million in grant funding. So the goal of the lab, and this is something I wrote in the first few weeks that I started, and it really hasn't changed since. It's our mission statement, so to speak. It's to establish a dynamic and productive research group with expertise in microscopy and studying caspase regulation in apoptosis, inflammation, and non-apoptotic processes. The ultimate goal is to understand how caspase pathways can be manipulated for the prevention and treatment of human disease. And a main focus is to create an environment that is conducive to learning and testing new skills. So this is Texas Children's Hospital, where I am based. It is affiliated with Baylor. So I work for Baylor and Texas Children's, and this is the building that I work in. Just a very quick journey throughout our scientific program. So I started with the caspase-2 activation reporter that I developed during my postdoc. And we moved on to kind of look into more what caspase-2 is doing. We were the first to show that caspase-2 is a tumor suppressor in a solid tumor model. We went on to show that caspase-2 can be activated in the nucleolus. And you see in this video here, caspase-2 is going to come up in yellow in the nucleoli of the cells. And we also have been looking at how caspase-2 regulates cell division. So caspase-2 knockout cells grow faster than the wild types. And this seems to be due to regulation of DNA replication forks. To expand the research program beyond just one protein, we also developed our techniques for looking at the inflammatory caspases. This includes caspase-1, and you can see a movie here, caspase-1 activation in green, followed by death of the cell. And we showed that caspase-1 and the related caspases, inflammatory caspases, are activated by heme that gets released from red blood cells. And here are primary human macrophages and the reporter being activated. So we're interested in this in hemolytic conditions such as sickle cell disease. And now we're at the point where we're using this information to generate and test some of our pathways in disease models. And eventually we want to move on to drug design to inhibit or activate some of these caspases. Along the way, I hit a number of personal milestones. I got married. I in the first year of my faculty position, had a baby, and he is now a 8-year-old mini human with his own thoughts and personality. So I just want to mention, about having a family while you're either preparing for or doing or during your faculty position. And maybe an uncommon opinion, but for me, having a child made me a better scientist, and for a number of reasons. I learned how to prioritize. I learned how to ask for help, which was something I wasn't good at before. I always felt that I had to do something in order to ask for something. And this way, but I changed my thinking to saying, in order for me to be successful, I need X, Y, and Z. I wasn't as nervous at giving talks, probably because I was too tired to care what people thought. I became much better at time management, as I had to leave at a particular time each day. And I became much more focused, both in my own work and in just the direction of the lab. So in the 6 months after maternity leave, we're hugely productive. I wrote and submitted an R01 from scratch. I submitted a paper, our second paper from the lab. I drafted figures of a second paper. And importantly, I cut a lot of the projects that weren't going anywhere and really streamlined the vision for the lab. and I'm not saying this just to boast, but it's just to combat people who say that women coming back after having children are less effective than they were before. I absolutely don't believe it. And this is always my story that I tell people. So I'm going to just spend the last while of this talk talking about what it is like being a PI. So in my mind, it's kind of like running a small business. You have to be a scientist, a manager, you manage people and funds. You're a teacher, you teach the people in your lab and elsewhere. You have to be a writer. You spend most of your time writing grants and writing papers. And in some ways, you're an entrepreneur because you have to raise the money to do your work, and you have to create a product, and that product is papers. And I also like to think of ourselves, think of science as a bit of an art to it, and especially when you get to make beautiful pictures using microscopes every day. So the truth of it is, like, we get formal training in one of these. If you're in a good PhD program, you may get some writing training, and similarly, you may during your postdoc or your PhD or postdoc, you may get some training in teaching, but it's not guaranteed. So you really have to kind of learn a lot of these things on the job, especially how to be this entrepreneur, and that is really coming up with the ideas for papers to get the grants and to produce more papers. So how do you get onto this cycle? Well, usually if you're lucky enough to get a good position, you'll have institutional funding in the form of a startup, and that'll set up your lab and fund your research for the first 3 years, cover some salaries. Institutes can also give you bridge funding if you don't get your renewal of your grant. And sometimes supplemental funding for lab supplies and equipment, but this varies from place to place. Some institutes like ours have pilot awards that you can compete for to start new projects. But once your startup, at least in the US, it's different in Europe, but once your startup ends, you're expected to cover a certain amount of your salary, and this is usually around about 50%. For the rest of it, you're dependent on external funding. And that's grants from government agencies like the NIH or foundations, American Cancer Society. And these cover everything you need, and they are highly competitive and peer reviewed. Federal grants are preferred by your institute because they have higher indirect costs, and also because institutional rankings in the country are based on only federal grants. So sometimes you feel like this guy here, just chasing money all the time. You spend a lot of time writing grants, but when you do get one, it does feel pretty good. So it can take multiple tries. In my case, it took 4 times to get my first R01, and I'm still working on the second one, and persistence is key. So if you get a tenure track position like I did, what does that mean? Well, it depends on the institution. And this is another reason why grants are important for promotion and for tenure. So some have renewable tenure where you renew it every 5 or so years, and some have, like ours, we have tenure track and non-tenure track positions, and there are subtle differences between them. But tenure is more of a prestige thing, and it helps you if you, for your reputation, if you want to go to work somewhere else. Most places have a tenure clock. Ours is 9 years. The focus is mostly on research, so you have to have a track record of obtaining grants, a number of peer-reviewed papers, at least half of them should be senior author, and a national reputation. What that means is that's measured by your invitations to speak in other places, letters of recommendation, etc. And sometimes you have to show a second item with, like, education, and show excellence in education, and service to the institute, which can be in the form of serving on committees, community outreach, and other things like that. So my advice, if you get a faculty position, start early when preparing for tenure. In your first year, you don't have to do much. You're focusing on starting your lab, but you do want to find out what the requirements are, what the timeline is, and are there different tracks? So in our case, we have to do 2. If you're a researcher, you really have to do research as your main track and education as your secondary track. If you do clinical work, then you can do clinical as your secondary or primary track. are institutional awards recommended, so we can get a education award, and that helps satisfy the criteria for education. What faculty development resources are there at your institute? You want to decide if you need to, whether you want to go up for tenure versus non-tenure, and what track you would go on. So as I said, research plus education or clinical, if that's available to you. So starting from the beginning, you really just want to keep your CV updated. Always keep emails providing feedback to you. So if someone sends you an email saying, hey, great talk, I learned a lot, just keep that in a folder. It's going to be useful to you. And the same with surveys, survey results for presentations and teaching things that you do. And if needed, you can make your own, but these can be included in educational portfolios and award applications to show your excellence in non-research activities. And also for your national reputation, build a network, attend meetings when you can, submit abstracts for presentation so you can get your name out there. You can get to know senior people in the field that could be possibly letter writers. And when needed, you can call on these people for favors. If you have a hole in your CV and you're like, can you consider me for this conference that you're organizing coming up or something similar? Teaching, we think of as being didactic, that we teach to a small group or in a lecture hall, but all these other things that we do as part of our research day-to-day is also counts as teaching. So teaching is a big part of our job. And so I just want to finish by just mentioning some of the pros and cons of becoming a PI, because there are many of each. You do have flexible hours. You can kind of come and go as you please, but at the same time, you often have to put in some long hours, and there is a huge commitment to what you want to do. For me, what I love is it's something different every day. I'm not just doing the exact same thing day in, day out. However, it does require an extended training time of at least a PhD and a postdoc, and sometimes PhDs are now requiring masters or a technician time before you even get into the program. One of my favorite things about the job is you get to be creative. You get to come up with ideas and test ideas and see if they work. But that has to be balanced with deadlines and competition and this overall fear of getting scooped, which, as I mentioned to you, isn't always the worst thing. So overall, I find it a very satisfying job, but also can be very frustrating at the same time, especially when you don't get grants that you spent a lot of time going into. And then one of the pros, if you're into it, is you get the opportunity to travel all around the world for conferences and such. But you may need to move to a new city to find job opportunities, which can be difficult, especially if you have a family. So if you are considering this kind of position, my advice going forward is, firstly, believe in yourself. I know it sounds hokey, but you really have to have inner faith in yourself to be able to persist in this job. Protect your time. Don't take on too much. Maintain a healthy work-life balance. It's way too easy to get sucked into your job and at the expense of everything else. And plan ahead for tenure, for papers, for grants. Each grant you should start planning it about 9 months in advance, if you can. Surround yourself with good mentors and trainees. Not all the advice you'll get will be good, but one of the great things about being a PI is that you don't have to take everyone's advice. But if you surround yourself with good people, they will be more likely to give you good advice. Be patient with yourself, with your trainees, and most of all with the institution and whatever bureaucracy you have to deal with, and be extremely persistent and try to have fun. So with that, I'll end, and I thank you for your attention. Here are our details if anyone wants to contact us. Thank you.
Thank you so much, Dr. Bouchier-Hayes, for sharing your journey with us. It really is an amazing journey. And, as someone who was a mom that did her postdoc, I really appreciate you being such an inspiration to mothers in science. So thank you. If you guys have any questions for Dr. Bouchier-Hayes, please input them in the Q&A section or the chat box, and we'll address them during the panel discussion at the end. So It is my pleasure to introduce our next speaker, Dr. James Quinn. So Dr. James Quinn is a translational scientist working in neurology at the Guided Genetics for Dementia Discovery site at Eisai in Cambridge, Massachusetts. He undertook his PhD at the University of Manchester, studying the role of tau in Alzheimer's disease. He then moved to the US and completed a postdoc in the Alzheimer's Clinical and Translational Research Unit at Massachusetts General Hospital. His postdoc involved studying the role of neuropeptides in Alzheimer's disease and TDP-43 in ALS. His research has taken him around the world and recently facilitated his move from academia into industry. So with that, I'll go ahead and pass it off to you, Dr. Quinn.
Thank you very much for the lovely introduction, and to Lisa for the very inspiring talk about being a PI. So let me just pull up my presentation. Here we are. Okay, so I think I was very well introduced there, but this is a kind of introduction slide. So let me move on. I've shared my Twitter account and my LinkedIn account if anyone wants to get in contact. Just a disclaimer, the presentation doesn't represent my current or past employers and is based solely on my experiences and opinions. So I made this slide for a talk I did about actually applying for postdocs about 3 years ago, and I thought I should re-include this. There are lots of alternative careers. I've bolded out the 4 that I have done or I've done in the past or currently doing. But I just thought this would be helpful just to get you thinking about, that not everything is either academia or industry. There is other things to those. And I know a lot of friends from PhD and postdoc who've gone on to be patent lawyers, to be editors of journals, to be even scientific illustrators. So there's really many opportunities that you get with having a PhD, and that's probably one of the most exciting things to happen about having a PhD. It wasn't in my introduction, but between my PhD and my postdoc, I actually worked in consulting for about 6 months as like an intern in a consulting firm, and it was a really good experience, but it definitely wasn't the right career for me. So that was a big thing in making me go back into kind of academic research. So I'll summarize my personal experience. So I did my BSc in biology at the University of Sussex. That's where I first got involved in Alzheimer's research, working with Professor Louis Serpell, looking at the interaction between amyloid and tau in the pathogenesis of Alzheimer's disease. And then I moved to the University of Manchester, where I studied, my whole PhD was on tau. And I was there from 2015 to 2018. And then I moved to Mass General to work in the Alzheimer's Clinical Translational Research Unit. So this is a picture of us all in the lab. I think this was a pre-COVID picture. And I was there from May 2019 to September 2022. And now I work as a translational scientist too, G2D2 in Eisai, and I started there in October, and I still work there right now. So I've just kind of broken my presentation into two or three main sections. So the first, I'll talk a little bit about how I landed my job in industry. I think Lisa really summed this up nicely about the importance of networking and attending meetings. It's how I actually ended up getting my job. So I attended a talk about new biomarker technologies in Alzheimer's disease, something that I was working on. And I met someone working at Biogen, and he gave a really interesting talk about Olink, which is a proteomic technique you can use to study blood and cerebrospinal fluid from patients to identify new biomarkers. And I just reached out on LinkedIn and said, would you be able to grab a virtual coffee? I think this was 2020 or 2021. So we just grabbed, a kind of 30-minute quick Zoom call where he discussed a little bit about his career to date, and I talked about my kind of interests. And that ended up then him suggesting that I talk to someone who recently moved from Biogen, where he was working at the time, to Eisai. And that was the kind of first link I had with Eisai. And I had another kind of virtual coffee with the guy from Eisai. And then I was going through my postdoc at the time, and I saw an invite to go out for this 3-minute flash talk competition organized by Research America. I was lucky enough to actually win it. And fortunately, it was judged by 2 people who work at Eisai, as well as 2 other judges. They were not in the research side, but they actually put me in touch with the head of the site here at G2D2. And I got to have a call with Jana, who is our site head. And I had that in September of 2021. So I think you can start to see a pattern forming. And then I saw an open position at Eisai. So I think, keeping an eye on multiple different avenues for kind of positions coming up. I was, looking in the Cambridge area and Boston area in Massachusetts. So there was lots of positions coming up, but I had some really targeted companies. I really wanted to keep working on Alzheimer's disease and in neurology. I saw an open position. I definitely did not meet 100% of the criteria, but they were looking for a PhD level scientist. And I kind of met about half the criteria, I would say. And then the person who I had originally chatted to, he then put me in touch with, he spoke to the hiring manager and then went through the referral. So I think that's a real key piece of advice. If you can get someone to refer you, that is extremely helpful. And, that can really, allow you to make sure your resume gets read. I don't think it guarantees you a position, but it definitely helps you get through to the recruiter. The recruiter's probably screening, especially in Cambridge and Massachusetts, there's hundreds of applications for positions. So I think, getting to be made sure that the hiring manager is able to see your resume. So generally, a lot of these kind of big biotech companies, they have recruiters working for them. So I spoke with a recruiter for the company, and then they put me in touch with the hiring manager. And then, so I interviewed with the recruiter in January 2022, and then it was followed by an interview with the hiring manager. And then it had an on-site interview in February. The on-site visit was actually very intense. It was 9 to 5. I had to give a 1-hour seminar on my projects to date, as well as having interviews and lunch with approximately 12 people. I'm assuming this is quite similar to what happens if you want to become a faculty member, but I actually found it was a really good opportunity to present my research and to a really focused audience as well. So I tried to, cover all of the main projects that I've been working on to date, and I got some really good feedback on my presentation, as well as on some of the projects that we were doing and potential routes towards publication. So I think that showed to me how much the science was valued in industry, and I got a lot of really good scientific questions. And someone actually I spoke to about this last week said he was sat in my seminar. He asked me a question about some of the statistics that I'd done in the work that I was doing, and I was very upfront and said I hadn't done the statistics. It was done by my supervisor. And I think the key here is you have to answer truthfully. You really can't get away with you lying and saying that you did something. I think you have to be really upfront about who did what, because there's real experts in the room, especially in industry. You really get kind of experts on data, analytics, and all of these things. So you've really got to be prepared to defend the work that you've done to some key experts. I was actually offered this and another role in March, and there was definitely a round of negotiation. And I've been told by recruiters in the past, you're kind of stupid not to negotiate. So there's always an opportunity to increase the salary in which you'll be brought in on, potentially get a signing on bonus, increase the amount of time off you get. And I was able to do that at one company and not at another. So there's definitely, different things that you can do. And I ended up accepting the role at Eisai because I really appreciated how much of a team mentality there was at the site. As I'm sure many people on the call understand about visas, I had to actually start my visa application in May. My visa got granted in September, and then I was able to start in October. I was able to do a change of status in the US. So I went from my J1, which is my academic visa, to an O1 visa that I'm using to work in industry. So I started in October, and you can see this is a very long time. So I think I started really looking for jobs in January, and then I didn't start until October. Yes, it was a little bit longer since I needed to apply for a visa, but I would expect at least 2 to 3 months if you were a US citizen, or if you're, applying in the UK and you're a UK citizen. But I think, you really have to think about these things way in advance. And, I can go back to 2021 where I first started learning about meeting these people. So I think really it was a very long process. So I was also asked to give a little bit of a day-to-day of what my work is like. So I kind of broke it up into similarities between my PhD and my postdoc. So I still get to work on Alzheimer's disease, which is the key thing that I wanted to do when I moved, from my undergraduate in Alzheimer's to my PhD in Alzheimer's to my postdoc in Alzheimer's. I really wanted to keep that through all the way into industry. So I kind of do general cell culture, stem cell work, immortalized cell lines, doing bulk RNA-seq, single cell analysis, running Western blots, FACS experiments, cell tracking experiments, using neuronal tracking and ICC, attending academic and industry conferences in person and virtual. I was at a Alzheimer's Association conference last week. So I think you can see there are a lot of similarities. I'm sure people who work in the lab do a lot of these techniques. So, there's a real expectation that you become an expert in these techniques. So you can really single-handedly just run it all on your own or with some level of support. So I think some of the key differences, I do have a lot more meetings. We're a global company, so I have regular meetings with the UK and Japan. We were just discussing before the webinar started about trying to get people from California and the UK. It's even worse with Japan. It's, I think, a 13 or 14-hour time zone difference. So there are a lot of, 7 a.m. meetings, 6 a.m. meetings, as well as some 7 or 8 p.m. meetings as well. There's a real expectation to present my data to the project teams every couple of weeks, as well as having real weekly check-ins with a manager. There's a serious emphasis on working as a team. You get really rewarded for that. So, I actually get individual awards if I've, really helped out on a project, which is really nice. And you get a lot of cross-training. So, if someone's sick, someone else can actually keep that work going. So I think that is a key difference to my PhD, where I was the only one working on the project, and my postdoc, where it was a very similar experience. Whereas now we're a team of about 12 to 15, all working together to really focus on, developing these drugs, and then in order to get them into clinical trials. So that's the end focus. And yep. So in conclusion, I think it's really important to embrace the unexpected, have multiple backup plans. I had another job offer on the table, so that was really important in case the Eisai job fell through. Take some time to decide whether industry is correct for you. I mentioned about postdocs. You can do postdocs in industry. We actually have a current postdoc, and we've had postdocs in the past who then got jobs in industry, but I also know postdocs in industry who then go back to academia. So they are real trainee positions. There are lots of alternative careers and similarities and differences between academia and industry. Some industry positions are never advertised. Recruiters can be your best friend. So I spoke to a ton of recruiters. Some of them were helpful, some of them were not helpful at all. And some still keep in touch with me. So, the whole thing never ends, really. You expect to be interviewed for the position no matter what you're applying for. Get everything signed and in writing. So I think, even from when I was applying to postdocs, I made sure that I got all the offer letters signed and, had them formalized. It takes at least 3 months to settle into the position. I'd probably say, when I first moved to the US for my postdoc, it probably took about 6 months to settle in. But in industry, you really hit the ground running. So within the second week I was here, I was in the lab. So there's a real clear expectation. You just get, you get in straight away. You're meant to be the real expert on the project. And just some, open news. We have open positions here, and there are lots more at Eisai. So if anyone's interested, go on our website. You should apply. I just thought I'd put them here. They're all based here in Cambridge. So I just thought I'd share that. And I just end with this picture of Boston I took. And thanks for listening. I've shared my LinkedIn and my Twitter again, in case anyone wants to get in touch.
Thanks, Dr. Quinn, for sharing your story with us. I really liked that you focused so much on, networking and then the negotiating aspect. I think a lot of people have difficulties with negotiating. It's difficult to sometimes know your own worth and brag about yourself. So thank you so much for that. If you guys have any questions for Dr. Quinn, please put those in the Q&A section or the chat, and we will address those during the panel discussion. So our next speaker is Dr. Harry Potter. Dr. Harry Potter is a teaching-focused lecturer in medical sciences at the University of Central Lancashire. His research interests focus on the neuroimmune developmental programming of neuropsychiatric disorders such as schizophrenia, particularly in the context of adverse prenatal environments. He is also heavily involved in student pastoral care and career development, as well as pedagogical research into modern assessment strategies. So with that, I'll go ahead and hand it over to Dr. Potter. Thank you so much for being here today.
Brilliant. Thank you very much. Can you hear me and see me all right?
Yes.
Brilliant. Okay, thank you to Afrida and Rebecca for inviting me today. So as Afrida said, my name's Dr. Harry Potter. The question everyone always asks is that your real name? Were you born before the books? And the answer is yes. And my students enjoy that. And it means they have no excuse to forget who I am. So I'm a teaching focused lecturer in medical sciences at the University of Central Lancashire, which is in the northwest of England. And I'm based on our lovely Burnley campus, which you can see is nestled really nicely in the northwest countryside. So I'm going to talk about my background and how that fits in with the sort of route I've taken through academia and how that's fed into my current teaching focus role. And then I'm going to end talking a bit about my current plans to sort of edge my way back into research, and then just some top tips that I hope people can hopefully use in their career paths. So my research interests are on the intersection between the brain and the immune system. So the way that external environmental stresses, things like maternal inflammation, experienced during pregnancy and then in the early postnatal period, how they interact together to affect our risk or resilience for chronic neurodevelopmental disorders like schizophrenia. And what I'm particularly interested in is this idea that an infection or inflammation during pregnancy can and will activate the mum's immune system, which is thought to adversely affect fetal brain development, and then, like I say, modulate our risk for developing chronic disorders in later life. So by all accounts, my route through academia was very traditional. Dr. Lisa talked a bit about this in her talk at the start. And I'm going to just talk through where I've come from and how that's led me to where I am today. So I'm from the southwest of England in a county called Devon, and I did my undergraduate degree in South Wales in Cardiff University. And during this time, similar to some of the other speakers, I got involved in some research. So I applied for some funding for a summer studentship and then elected to do a placement year. So I was working on rodent models of neurodegenerative and neurodevelopmental diseases. And that really got me interested in research and inspired me to start looking into PhDs. So as I worked my way up through the academic route, I also worked my way up through the country in England, and I moved to the University of Manchester, same as James, to do my PhD, working on those rodent models of maternal inflammation. And during my PhD, I had a lot of teaching experience. One of the really valuable parts of my PhD was being able to interact with other postgraduate students and teach undergraduate students. And that was definitely the part that I really enjoyed alongside the lab work as well. And at this stage, when I finished my PhD during the pandemic, I really thought that my path would be to carry on and continue down this traditional academic trajectory. So I applied for a postdoc and started at the University of Edinburgh, working in a fantastic new lab with really supportive mentors, really great facilities at a university that had a really high prestige. And where my route sort of differs here is that I actually ended up leaving this postdoc after 6 months. So 6 months into my 2-year contract. And that was a really difficult decision at the time, again, because I had really supportive colleagues and supportive mentors. And so breaking that news to them and saying that I was going to leave so soon into the postdoc was a really difficult decision to make. There were a couple of reasons behind this. One of them was that I actually really missed the teaching side of my job previously. And whilst this postdoc position was great in terms of the research program, it was a bit more limited in terms of how I could teach students both in the lab and in lecture theatres and in other routes as well. So I actually ended up leaving this and moving back down to the northwest, starting a job at the University of Central Lancashire, which is very close by to Manchester in the northwest of England, and that's where I am today. But in preparation for this talk, I was reflecting on some of the pros and cons of those two different routes, so the teaching or the research route in academia. And so that's what this slide is going to detail. And this, again, is picking up on some of the similar things that other speakers have talked about already today. So obviously one of the great things is being able to move locations and experience lots of really interesting and vibrant communities and cities. And I've been really lucky. 3 cities that I've lived in the UK are really great places to live and also are close to lots of nice countryside to allow you to explore the country a bit. Working at more traditional, we call them Russell Group universities in the UK, means that you're exposed to lots of really varied and interesting expertise. So people that have really relevant research interests that can help to support you and guide you through those programs of study. I've also really enjoyed and appreciated the opportunities to travel, so to present my work at conferences, and the neuroscientists on the call will know about FENS and SFN as two of the major international neuroscience conferences. So I've been able to travel to them, which has really enriched my research and allowed me to network with people outside of our lab and outside of our country. And then the PhD and the academic route of research that I've taken has also allowed me to kind of access other opportunities outside of the university in the wider community. So a couple that really stuck out to me and have really sort of molded my career development are being able to present my research in the UK Houses of Parliament. This is me next to my my wonderful local Labour MP, Jeff Smith. So I was able to go to London and present my research to policymakers and parliamentarians and present my work to a completely different audience. And then on the teaching side, I was also really fortunate that my PhD supervisor was an expert in animal behaviour and asked me along to teach on an animal behaviour field course out in South Africa. So I think these extra opportunities that a PhD and a career in academia and research open up are really important in shaping any career, regardless of whether you go into research or teaching or otherwise. And then on the flip side of that, there are always the negatives that come alongside that. The moving, which has been mentioned before, again, having to move every few years as and when your post ends or when a new project comes up. Whilst that can be great to experience new places, there is the burden of doing that, of leaving your family and your network. One thing that really was important in my decision was this idea of job security. So the thought of having to potentially work in several different roles where I was on fixed term contracts for a couple of years at a time and having to always be on the lookout for where my salary was going to come from. And then the final thing was the workload, and this is not necessarily the amount of work that a research career had, although it is a very intensive job, but more about the split of the workload between the different activities. So I really wanted to put more of my time into teaching, and the previous job, like I said, didn't really allow that. So that was one of the factors that encouraged me to start looking for a new job. So this brings me to what I'm doing now. So as I said, I'm a teaching focused lecturer in medical science at the University of Central Lancashire, and I work on our Burnley campus, which is a very small and new but rapidly expanding university campus based in the northwest of England. And you can see a few images of that here, again, nestled in Burnley, which is a small working town in the northwest, but adjacent to lots of lovely countryside. So it's a really nice environment to work in. UCLan is what we call a post-92 university in the UK, meaning that it's much less research intensive. So whilst there is fewer opportunities to engage in research, it means that there's a much more heavy investment in teaching, and particularly in widening participation. So encouraging students from the local area that wouldn't normally engage with higher education to come in and learn on our courses. One of the challenges of my job that that drew me to this and that I really enjoy working with is that we teach our degree over 2 years. So rather than the standard 3-year undergraduate degree, we squash that all into 2 years on an accelerated course, which comes with more challenges of having to work throughout the summer and teach throughout the summer and get the students through a lot quicker. But what it does mean is that we encourage students, again, who maybe wouldn't want to spend the 3 or 4 years doing an undergraduate degree to be able to come in and achieve that. So we get a very different range of students who bring with them a lot of diverse backgrounds, both educational and social. So my role primarily is as the module lead for an immunology module, so it aligns very much with my research interests. I'm also the year 2 tutor, so this is a role that I've picked up throughout my time here. This really involves the pastoral care of a whole year group of students and making sure they're all essentially okay and can study and are able to finish on time. And then another aspect of this that I've elected or picked up on my way is acting as the academic lead for what we call the Scholars Program here. So this is a widening participation public outreach program where we enroll local high school students, again, who traditionally are from families or backgrounds that wouldn't normally engage with higher education. We enroll them on a program of engagement activities and research tasks to get them interested in studying medicine and also other degrees in higher education. In terms of what my typical week would look like, obviously a lot of that is delivery of content. So I think in my contract, I'm actually legally not allowed to do more than 18 hours of face-to-face sort of primary delivery of content. But like I said, this is really molded around my research interests. So whilst I'm the module lead for immunology, I'm also able to teach hematology, teach neuroscience, lab skills, molecular biology, and then supervise students throughout the degree on their dissertation research projects. A big part of my role is that student support. So in terms of their academic support and attendance, but also a sort of unexpected part of my job that I really enjoy and really have great satisfaction in is supporting students as a whole person rather than just an academic or a number on a spreadsheet. So our students, like everyone, come in with lots of different backgrounds and often have lots of emotional and psychological issues that will come up as they study on their program. So a big part of my job is making sure that these students can get the right support they need and signpost them to the person that's qualified to do that. Again, similar to what Dr. Lisa was saying earlier, we don't necessarily get trained specifically in all of these areas, and it's something that you have to pick up on the job. So it can be quite stressful and emotional to make sure that you are providing the students with the right kind of support. But like I said, it's a very satisfying part of that. One thing that I was warned about when I was taking this job from my research colleagues was, oh, there's going to be lots of admin on that job and it's going to take away from all the other things you like doing. I'm one of those people that actually really likes a spreadsheet and likes data and getting everything together. So a lot of my job involves making the content, marking and assessment, and making sure that what we're delivering is of high quality. And then after all of that's done, and maybe in some of the holidays, the academic holidays that we have, I do have time on my workload to engage in what I've called all the other academic fun stuff. So things like public engagement and outreach, and also research, which I'll come on to on the next slide. And of course, all of this comes with quite a strong dose of imposter syndrome, and particularly my family and friends will have commented at the start saying, don't you get scared standing in front of 100 people and talking and delivering a lecture? And I think this is just like any aspect of imposter syndrome. You learn that you can actually do the job and you are qualified to do it. And it gets progressively easier to stand up in front of a whole group of people and deliver a lecture. One thing that I think is really interesting about my journey is that when I said to people or told colleagues that I was going to leave my research job and move into a teaching focused job, is that I got a lot of feedback that I put in inverted commas here, which at the time felt like I was being told that it was the wrong decision. But looking back, it's probably just people guiding me and telling me what could happen as a result of this. So I got told things like, don't you know you're giving up research for good and you'll not be able to go back into it? Or don't you know you won't ever be able to supervise PhD students again? And what's really satisfying and enjoyable about my position at the moment is that I'm able to focus the majority of my time on teaching from the comfort of a permanent job without having to worry about short-term contracts. But I am also able to keep up with research and carry on publishing from previous roles. And I'm now at the stage where I'm looking to try and seek some internal and external funding. So within UCLan to try and get some PhD students to start up a lab with some collaborators, and then also looking at external funding from some of the big funding bodies in the UK. One bit of advice I would say for this is that if you're working at a university, they'll almost definitely have an internal grants and funding unit, which I never knew about before until I came to UCLan. So this is a resource that's really beneficial in guiding me through this process, all the way from thinking about applying to actually putting the proposal together, and then all the way afterwards as well. And one thing that I'm a big advocate for is, again, like previous talkers have said, is maintaining your network and reaching out to people that maybe you only met at a conference 10 years ago for a couple of minutes, but you chatted to them and you have some shared research interests, as well as reaching out to new people, just that you find that work in the same area as you. And what I find is that even if half the people don't ever reply to you, the responses that you do get are almost always positive and lead to some sort of collaboration, whether that's all the way up to a big project or just a collaboration online. So I'll just end by presenting what I think are some of my top tips. And I put this caveat here just to recognize that I am very fortunate in the job that I do. And I also do not face a lot of the same systemic barriers that some people or a lot of people in academia do as well. So these top tips are here from my experience, whilst recognizing that in a lot of cases, there are other things that are getting in the way of some of these aspects. So the first is to always seek out opportunities and just keep open minded that these can arise in the strangest of places and not necessarily land on your desk. You might have to go out and find them. The second is to make sure you're always connecting and make use of that highly valuable network. Again, even if it's someone that you just met for 10 minutes or you connect with on Twitter, it's always worth just reaching out and getting a feel for what's happening, because that can lead to something really quite good. My third is really relating to my sort of career choices, is to not be afraid to break away from what some people might be telling you about what the traditional or standard path might be, and just to take that jump if you feel like it's the right thing for you in that moment. And then lastly, just to prioritize. So first of all, what's important to you within your job? So for me, it was the teaching over the research, which is why I decided to go down this path. But then also prioritizing what's important for you outside of your work life, in life generally. And whilst it's great that a lot of people in academia and science are really passionate and like to give more time to their job than they are contracted to, just that a reminder that a job is just a job. You're contracted for a certain number of hours per week and you're paid for those hours, and there is nothing wrong with just doing those hours and living your life outside of that. So the work-life balance is really important there. So please do connect on Twitter or via email if you want to reach out and have a chat, and I'll be happy to answer any questions about this sort of career path. Thank you very much.
Thanks, Dr. Potter, for sharing your journey with us. I really appreciated you talking about, the student care. These are types of things that you don't necessarily see written out in the job description, but are definitely an important part of the job. So thank you so much for that. Our next speaker is Dr. Vrutant Shah. Dr. Vrutant Shah did his PhD in cell and molecular biology at the University of Houston, where he explored the gene regulatory network and mRNA regulation in fate specification of the ectoderm. He then did a postdoc at MD Anderson Cancer Center, where he developed a preclinical mouse model for metaplastic breast cancer and determined therapeutic targets that can be explored further in clinic. Currently, he is working as a research associate 2 at Methodist Research Institute and developing mRNA techniques to target metaplastic breast cancer cells using RNA editing enzymes. So with that, I will hand it over to Dr. Shah.
All right, thank you so much, Frida. And I'll share my screen. All right, so thank you for the introduction and good morning, everyone. So I'm gonna talk about my journey till what I have become, the role I'm at the Houston Methodist Research Institute. So my love for science starts, as all the speakers have talked about, but in India, where we grew up with a very limited exposure to science, I would say, when at least I was growing up. So I attended a science fair and I participated in the science fair, where I was doing some, basic plant experiments in my house, and my interest was mainly in the plant genetics at the time. And of course, being in an Indian culture, your parents' expectation is to become a computer engineer or a doctor or something like that. And I was not one of those. So of course, I had to fight a lot with my parents for my research experience. And luckily, I participated in a student exchange program, which was in USA, where I stayed 6 months with a Amish family. Luckily, they picked me up somehow. And then another 6 months, I stayed with a proper American family. But in both the cases, I studied I studied high school here in my last year. And I had an amazing bio teacher. Her name was Miss Kingsley. And she gave me plenty of opportunity to expose myself to different kind of sciences, molecular sciences, genetics, biochemistry, and everything in between. And I actually participated in summer workshop at Indiana University, and that just blew my mind, like how much I can do with science, and I had never experienced that. So I applied here, I got admission, but luckily I did not get the visa to study in USA. And so I said, okay, what's the opportunity now in India? So I did my bachelor's in India in Bangalore University with the 3 different majors, zoology, genetics, and biochemistry. It was an intense coursework. It's not like the US system where you have the labs and the lecture 50-50 kind of. But in India, the lecture portions are like heavily. So you have to be just studying from the books most of the time, and the labs is just addition to it. After that, I did the industry experience for a year, where I learned how to do the protein chemistry, where I did the manufacturing of cancer recovery agents and protein purification. And I was also team up with the R&D, because that's where my research interest was, to scale up the manufacturing from making like 1 microgram of protein to 10 microgram of protein in a kind of a GMP setting. So after that, because my love was always to study abroad, so I came here as a postbaccalaureate student, studied 1 semester in Iowa, and then transferred to University of Houston. The main reason was to transfer is because, you know, the Midwest is mostly the plant. And at that time, my interest was already shifted towards more molecular into the mammalian system. So at the time in the University of Houston, I got an opportunity to work with Dr. Roman. He was studying a Drosophila genetist and in a behavior and molecular biology. And what interested me was we were feeding Drosophila, or giving a choice rather, to have one as alcohol and other one as a sugar. And we were seeing how flies are selecting between alcohol versus a sugar. And that's called the propensity experiment, where you're like asking flies to basically, are they addicting on alcohol or not? It's same as the human. You know, once you have alcohol consumption every day, you start building the addiction. And that was our studying, and that was very interesting. And unluckily, he had to move after 2 years to a different institute. So I could not carry forward this research, but I was mainly interested in the molecular functionality in the brain, how alcohol changes that. So next, I decided to do PhD, and my wife moved to USA as well, so I could not leave the state, and I had to be in Houston for a while, because she was also into PhD program. So I decided to do PhD in developmental biology, but looking at the molecular and bioinformatics side. So my main goal was to identify how the ectoderm, which is one of the basic germ layer, decides to become epidermal ectoderm and neural ectoderm, while shutting down everything, all other cell types. And as we know that during the transition phase from maternal to zygotic transcription, there is not much of a protein in the play. Mainly there are microRNAs. And so what I did is I explored the role of microRNA by clipping the Argonaute 2, which binds to microRNA complexes, and also doing the poly A and ribodepletion mRNA, and gathering all this bioinformatics data and to understand at the gene regulatory network side that how all this play in a role during this very 30 minutes time scale, because from ectoderm to epidermal or neural ectoderm is decided within 30 minutes to 1 hour in the xenopus embryo or the egg. So I did that, and I published a few of the publication along the way. But the major part what I learned in my PhD was exploring the unknowns, because at the time the Xenopus transcriptome and the genome were in their early phases, and the mouse genetics and the human genetics were way ahead of the time, and there are not many tools to incorporate the unfinished transcriptome into the research. So I learned a lot of bioinformatics pipeline. I learned the wet lab, how to manage the wet lab and the dry lab. And I think that's the skill now everyone wants to do in their life. And I was learning that during like 5 years back. So it was difficult, but it was manageable. And I had a great support from my mentor, because she sent me to the workshops. She sent me to the people who knew how to do this thing. And my job was to bring it back to the lab, try to analyze, try to play with it, and see if the data makes sense. And I had a great thesis committee where I tried to have 3 bioinformaticians and 3 wet lab people who can give me the feedback on my PhD commitment. And that worked out really nice. During my PhD thesis, I also developed the sidetrack where we were looking at the post-traumatic injury and how the microglia and the astrocytes play a role into recovery of this injury. So that's something the lab took forward, and I decided I had enough of developmental biology. I want to have more translational experience, because this is all basic biology, and I did not see an output to the human world. So I transitioned to the cancer biology down the street in Emily Anderson. And the reason I picked the lab is because, again, the work-life balance, because the wife is still in the PhD program and you have to be in the town. And I said, okay, yeah, maybe for 2 years we'll stay together while she finishes her PhD, and then we'll go wherever the destination takes us. So I thought of a quick postdoc. And so at the time, Dr. Michelle Barton, she was developing this new breast cancer model, the metaplastic breast cancer model, by overexpressing one of the protein called TRIN24. And what metaplastic breast cancer is, it's a very typical and it's very rare and aggressive breast cancer in TNBC, for which we don't have a treatment till now. The reason it's very aggressive and hard to treat is because, as you can see here, that it has 2 different cell types in the same tumor. So you have the epidermal cells and also the mesenchymal. So the stroma-like are the mesenchymal and the islands are epidermal. And our mouse model very much represents the human condition, where also you have the epidermal and the mesenchymal cell type. So it's a very hard disease to treat, and luckily we had this model where most of our tumors were metaplastic. So that got me intriguing into this model, try to understand its molecular behavior and how exactly this tumor are arising, and what can I do in order to come up with a treatment plan. So it was very fast pacing, because we had to come up with some clinical targets, and also very challenging, because to dissociate this tumor, to get this tumor into the immune background, growing it and trying different drugs on it was very challenging because of the mouse genetics, because of the challenges from the regulatory authorities, and also working with the clinic, because the clinic is always like, did you find something? Did you find something? And I'm like, dude, it doesn't work like that. You need to be patient. I need to analyze the data. I need to be sure that it works in cell line, it works in mouse, and then I can come up with something. But after work of 4 years, I was able to publish into Nature Communication. And it's a big collaboration between the clinical world, also the mouse world, and also the people in between, where they take the clinical samples, make the PDXs, it's called the patient-derived xenografts, and then proving those xenografts also had the same molecular characteristic as my mouse model, and come up with a molecular mechanism and the target to see that whether those can be taken to the clinical. So after my postdoc, I was in a dilemma because my boss moved to a different institute, and I really want to do this research further. and there are not many people who does this research in the Texas Medical Center. There's only one person at Methodist. She was doing this. She's still doing the research in metaplastic breast cancer. But I was looking at the industry. I was looking at my visa option. I just applied for my EB2 category, and I-140 was approved. And I got the opportunity in a startup in California. They were offering me O1 visa, and my wife is working in a clinical research here. And I'm like, shall I go take the O1 visa, or shall I be in the Texas Medical Center, find a lab where I can still do this metaplastic breast cancer research? And luckily, I got a call from one of my friends, and she said, hey, the lab you're talking about, Dr. Chang's lab, one of the collaborator is going to start up his own lab at Methodist, and would you like to have an interview with him? So I said, okay, sure, why not? And so I got a call on, I applied to the same position in the 2 different labs, and they both in the same institute. So they both co-interviewed with me. And at the end, since the current PI, Dr. Christopher Brannon, he's also going to work on the metaplastic breast cancer. He, like within 30 minutes after the call, he sent me the offer letter. And I was like, wow, that's quick. Shall I still look for some other opportunity, or shall I go ahead with this? So I said I'll take a day or two to decide whether I want to go with this. But the options were very limited. And I'm like, this is a really good position. I can still write my own grants. I can still apply for K22 in future. I can be still on an academia track, and I can learn the new technology, because his lab is all about technology. So I'm like, I'm bringing the cancer experience. I'm bringing the model experiences, and now he's gonna teach me the techniques. I can combine both of them and come up with a really good proposal. So I said, okay, I'll go with that. Here at the Methodist, currently where I'm working as a research associate too, we are into the mRNA vaccine and the development of a new mRNA technology. And I'm learning new technologies like eClip, then the RNA sensor, which is an upcoming field where you're trying to target the mRNAs by having a stop codon in your transcript. And once the RNA editing enzymes recognize the target and your transcript, it turns on the stop codon into the the turn on transcript, and you can have the downstream payload, or whatever you put as a killing agent, can be activated. So we're working in the very early phases of that, but the opportunities are great, and I really enjoy my work. It's the same as our PhD and a postdoc, but I had to do a lot more because I had to, being a senior most in the lab, I had to work on the regulatory aspect. I need to mentor all the people comes in the lab. Also, I had to be into the lab lead duty. Basically, that means that I had to manage the lab, manage all the aspects of the hospitals and the regulatory side of it. A lot of paperwork, a lot of meetings, but I still enjoy my work every day because I learn different stuff, and I still be on the metaplastic breast cancer research, which is my goal to come up with a more clinical options so that people can have a clinical trial. So I'm very much fortunate to have a great mentors in my life. So that's my PhD mentor, and that's my postdoc mentor, and that's the group I work for last 10 years. And during my postdoc third year, I had a baby, and I was writing K99. My boss had to move to a different institute, and we had to drop the plan for K99 because I needed to have a dual mentorship. And it was a COVID time. It was a bit hodgepodge at the time, and I could not finish my K99 opportunity, but I was determined. And so I said, okay, I need to have a grant, and I want to try at least once to be on an academia track. So I'm still hoping that in next 2 years, I will be able to write K22 and have my own lab someday in future. So what I learned throughout my journey, that I usually go with the flow. And when you are desperate, the opportunity comes to you, and don't say no, I guess. Network, because many people I know from my PhD and postdoc life, they end up in industry. And whenever I have question, I just give them a call. They hook me with somebody they know, and that's how my network expands. I already talked about the opportunity when it comes to you. Learn the technique and master them, because that is the key to success, what I feel. I learned bioinformatics, and as you know now, the bioinformatics field is exploded. The single cell RNA technology and the new clip technology, RNA sensor technology, they all require some bioinformatics expert, some wet lab experience as well. So combining both of them, I feel, is the best way to go. And always I'm looking for the better role, even within the institute. If I get an opportunity to become research scientist, I would not say no to that. And always keep your doors open and never burn any bridges, because you never know who is helpful to you. And of course, work-life balance is important. I do understand that I had a kid and there was a rough time for me, COVID, and I had to let the K99 go partially because of that. But I'm not regretful. I'm more determined to get K22 next. So it's motivation. He's a love for life, and he always motivates me to do something better. So that's my take on to the journey. I hope you enjoyed my talk. Thank you.
Thanks, Dr. Shah, for sharing your journey, and also, giving the tips, and also, sharing your obstacles too, especially, some obstacles that are cultural. I think these are important things to also address. So thank you so much. If you have any questions for Dr. Shah, please put those in the chat or the Q&A box, and we'll address them at the end during our panel discussion. So our last speaker today is Dr. Rwik Sen. Dr. Sen is a field application scientist at Active Motif. As a field application scientist, Dr. Sen educates researchers in academia and industry on Active Motif's technology, develops new applications, builds alliances, and brings in revenue. His training at Southern Illinois University and the University of Colorado, working with an inclusive group of mentors, and his own experiences with mentoring and communication contribute to his ever-evolving role. So with that, I will pass it on to Dr. Sen.
Thank you, Afrida, for the introduction. Thank you, everyone, for attending. I'd like to thank Proteintech for this opportunity to present today. Is my screen visible?
Yes, it's visible.
Okay, thank you. So my name is Rwik Sen, and I'm the field application scientist at Active Motif, and here is my LinkedIn. Feel free to connect with me. I'm currently based out of San Diego, California. So my career trajectory is shown here. I completed my undergraduate degree in 2009 from India, and then I came to the US for my PhD at Southern Illinois University. In 2013, I got married, and then in 2016, I went to the University of Colorado for my postdoctoral training. Two important events that happened there were my postdoctoral fellowship from American Heart Association, which also helped me with learning grant writing skills, and then getting selected for a course at the Marine Biological Laboratory at Massachusetts for some time. And then there I got to meet a lot of people from across the world who came as students as well as faculties to take different steps of that course and workshop. And then I got this job at Active Motif in 2020 as a field application scientist, and I just completed 3 years there. So that's been very exciting. So as all the other previous panelists have spoken, I wanted to highlight here that I got married in the middle of my PhD. And as you all know, that PhD is not an easy journey, but getting married really helped me, because there were moments where you really need somebody to be with you and to support you through this ups and downs of your journey here. So this is my acknowledgement slide, which I'm showing early ahead in my presentation, because I don't want to acknowledge people who helped me get here at the end. So there have been several people, my mentors and peers, who have supported me at each stage of my career. They themselves come from diverse career fields and career levels. Not necessarily everybody has to be a PhD or an academic professor to be your mentor. Then they come from different organizations, and spouse and family support was absolutely necessary for my success here. Then you must have work-life balance. So to get work-life balance, I would go out, hang out with other students, and we would do fun activities together. So all my professors and mentors from my undergraduate, PhD, postdoctoral training, and my mentors at Active Motif, I'm acknowledging all of them. The next interesting thing is training. So for my training, what I did was I chose to go into a new field at every stage of my career, but I was also able to contribute the skills which I had from my previous training into my new role. So if you'll see here for my PhD, postdoc, and the current job, the main central theme is epigenetics, so I could contribute there. But also, I learned new fields in every step. So in my PhD, it was transcription and DNA repair. Postdoctoral was developmental biology. And in the current job, it's novel high throughput assays. A lot of different kinds of assays, which you can see in the Active Motif portfolio if you go to our website. And you can also see the different model systems and methods that I learned at every step. Then comes a very important part is communication. You have to be very good at communicating. And this is something I'm still learning every day. So you have to be good at building and maintaining alliances, connecting with alumni. Maybe there are alumni who you haven't met in the past, but you can ask your mentors and your professors to get you connected to those people. And then communication, to communicate the significance of your work, because we often spend long hours and very hard work on doing a project. But when it comes to communicating, we don't necessarily communicate the way it should be or the way it should make an impact. So we must know the significance of the work. Not everybody works on developing drugs that will directly cure somebody, but all of us do something which is significant. So we must appreciate that. And then you have to be prepared with a customized speech to tell, like, according to the nature of your audience. Your audience can be people who are strong scientists or people who doesn't know much about science. So you have to be able to give them your elevator pitch, which is a short pitch of what you do. Then there are several events and organizations who promote academia industry alliances. You'll find them at your home institutions. The University of Colorado, where I did my postdoc, was very good in that. So I was part of the Postdoctoral Association, and these alliances, we organized conferences. That gave me a very big platform to prepare myself for my current role. And then being able to present at national conferences, getting awards like the AHA Fellowship or the Peer Supporter Award, those are, again, very big things to boost someone's confidence and propel them forward. So I have also been supported by other things like peer-reviewed publications, but this is something very interesting because not every industry role requires you to have publications. So it's very role specific. And at the time that I was applying, I did not have these many publications or this Google Scholar metrics. So this is something that has happened quite recently. But at Active Motif, what I try to do is organize my peers together and go for publications. We've done reviews and book chapters. So whenever a good reputed journal, not the predatory ones, so be careful of those, they reach out to me asking for something, I try to help them. Then I did a lot of teaching during my postdoc, and there I learned about course designing and other techniques of pedagogical training, which helped me give presentations and is helping me in my current role. I did a lot of mentoring, and during this process, I learned and understood about the importance of diversity, equity, and inclusion. And this is something I carry with me ever since. And this is important regardless of what field you're studying in and what kind of job you're doing. I helped the graduate school with presentations during orientations and other events like recruitment, tutoring. Those were all very helpful. And finally, peer reviewing and judging at national conference and local scientific events, again, helped me realize the kind of work that I would like to do and these are also very helpful to put in your CV for recognition of you by others as a scientist. So in my current job, I'm helping Active Motif with this capital equipment, which is called Pixel Multi-Sample Sonicator, and it's being used by people in all of those fields, as you can see listed in that box. And the reason I put the pricing here is just to show you the importance and the impact it has on the company. So this instrument is new, and I am hired as the field application scientist to educate people on this instrument and also to work with different departments of Active Motif to get this unit from the factory to the customer and continue the customer support throughout the lifetime. So the work involves a lot of communication, active learning, because I haven't learned everything about this instrument from literature or from books. I have had to learn it just by communicating with different people. Teamwork, meeting new people, managing expectations, timelines, and costs. So one part of my job is product management. All the duties are listed here. I'm happy to talk about them in details, whoever wants to connect offline after this. Research in the lab, because we constantly need to develop this new applications and everything. And to do that, I have to work with all of these people externally and internally at Active Motif on a daily or on a regular basis. The other part of the job is very customer facing. I have to do all of this, quality control, including working with these organizations for regulatory things, logistics, legal agreements, shipments, and again, working with all of these people at Active Motif and outside to get through all of these things. Then finally, I'm always seeking new opportunities because I want to grow and learn in this new role and not just be confined to this one role. So I do support the R&Ds, do some lab work, develop new assays. I'll be presenting a poster at AACR in Orlando, Florida next month. So I'm excited for those of you who are going there. And then we submitted an SBIR grant for this, so continuing my grant writing skills. Services lab at Active Motif, where customer services come in and we deliver results. So single cell assays are something new that we developed. So I was part of that, where got trained and did some experiments. And then territory management. So international territories, some of those countries I manage, and I was able to sell this instrument there. And this is something very new to me. This is my first job in industry. I never worked outside of academia before, never had any gaps. So I am really happy that Active Motif is so supporting in equipping me with all the skills that I need to do this job. So applying for industry positions requires a lot of time, research, and communications, and I'm going to share some of my experiences that helped me get here. First, you have to understand the importance between CV and difference between CV and resume. CV is your master document, which you update chronologically. You may need it for an academic position, but not for an industry. In industry, you are asked to submit a resume, which is a one-page quick read, which you tailor according to the job advertisement, taking information from your CV as necessary. I have seen recruiters firsthand spending just a few seconds going through the resume, and that's it. So your keywords have to catch their eyes. Second, submit your resume, sign up for job notifications in as many job websites and company talent pools as possible. They will reach out to you if there is a match. They usually use algorithms for keyword matches, so be careful in choosing your keywords. Be very careful for scams. You must be following the news. There are lots of fake things out there. People should never contact you from their personal emails, never ask for money or your ID. They will only ask for your social security or your ID once you got the offer and they're hiring you right there. So you'll know when it's genuine and when it's fake. LinkedIn profiles are very helpful. You can mark yourselves as looking for a job, and then people can reach out to you. I have been contacted by several recruiters, and they usually do that by searching your about section or your resumes and screened by keywords. Then hiring for industry positions occur very quickly, unlike academic positions or postdocs. So you have to be ready to make the transition within a few weeks to a month when you are actually talking to somebody, or make it clear at the start that when your timeline is. Then you have to be very good at communicating, and you can start this, if you're an introverted person, you can start this by communicating with seminar speakers who are professors from other institutes coming to your home institutions. You can go to vendor events, communicate with those people at your home institution. So these are some things you can do right at your home institution. Then there are almost all institutions have some academic industry alliance events, or if it's not in your institution, there might be things going on outside, so you can participate in those. Some people get to go to conferences. I realize not everybody has the funding or ability to go, but conferences are the best places to network. Then you can always reach out to scientists whose publications and protocols you have been following. Their corresponding authors, their emails and everything are listed in PubMed or their publications, so you can easily find them and contact them. Then you can also take part in industry events, or just sign up and be an audience with events hosted by industries. For example, Active Motif, if you go to the resources webpage on our website, you'll find the Chromatin Club, several blog articles, podcasts, and our annual event, ChromatinCon, which are where big scientists from across the world are lined up throughout the day to give presentations. So you can network with people through all of these channels. And I'm sure a lot of other companies have these kinds of resources, including Proteintech. So then you can also use appropriate social media channels to reach out to people. LinkedIn is more focused to industry, but industries also have a Twitter presence, and a lot of labs and faculties today are on Twitter, so you can reach out to people there. And you can also work with companies who have a long-standing working relationship with your lab. You have those representatives coming in to sell products and everything, so you can talk to them, ask them to connect you with the appropriate department at their organizations, and that way you can build relations with them. Then you can also work with companies who have new products and they want somebody to test them, validate them, so you can, and you give them the results in exchange, which they can put up on their website. So these are all different ways you can communicate with people. Always be prepared with these two main questions: why you are interested in that specific job and that specific company, and if you get the job, what are your short-term and long-term goals? Because these are questions almost every job interview you're going to be asked this, and this is something you can develop over time. You can polish it by practicing with your peers, with your mentors, so that when you are in that actual real position and being asked, you give a very good convincing answer. So I'll take the next few minutes to show how you can navigate the job and build a resume. So every company will have something on their webpage to take you to the careers page. For example, Active Motif and Proteintech websites are shown here. They have these About Us tabs where if you click, you'll go to the careers page and you'll find the kinds of positions listed. The position types can be full-time, part-time, work-study opportunities, internship. So full-time positions also include industry postdocs for some companies. Work study and internships are different because internships are generally temporary. Work study is something where a student can also work and both go parallelly. Then there are categories like on-site positions, remote and hybrid positions. You can choose as necessary. So how would you build your resume? That's a whole topic for a whole other seminar, but here I'll just show you one quick skill. There are things called transferable skills, which are skills that you have but not necessarily listed in the job posting, but you'll have to identify them from the job advertisement and see what skills you have that can match that. So some of those are like your communication skills and experiences and expertise, which you can draw from your training. So I'll use one old job from Active Motif website and show how you can do that. So this is a listing for a certain job that was there. And one of the things here, it says present results internally. Now you may have never worked in a company like me, and you may, so you don't have the experience in presenting results internally at an industry setting. But how will you prove that you are still qualified for this? You'll show all of your communication skills and the things that you have been constantly presenting research at your departmental institutional meetings, lab meetings, and conferences. The other thing is project management here. This is a paid certified course people get for project management. Not all of us have this certificate. So we can draw from our experiences of how we do our projects and meet the timelines and funding and everything and show that we are qualified project managers, although we don't have certification. Team contributor, we may be individually working on our projects as PhDs and postdocs, and we don't necessarily work in teams like in an industry. But again, at the end of the day, if you look around you, you realize that you are still working on teams, because when your paper gets published, you almost always see that you're not the only author with your PI. There are other people in the paper, so you just have to recognize that. So hiring has some general steps. The most important things you need are confidence and communication. So the first thing that you would need to do is spend time, research the job and the organization, and then, if possible, do an informational interview. Informational interview is asking someone for a few minutes of their time to know about the company and the role that you're interested in. So someone from that company. Now, you may not know somebody in every company you're applying to, but you may have second-degree contacts through LinkedIn. So you ask your primary contact in LinkedIn to connect you with that person and set up a meeting there. And always reach out and acknowledge that person for their time. Then you apply with a tailored resume using keywords, and you follow up if you don't hear back within reasonable time. I've been told personally by several recruiters that even if there is a 40% match between your skills and the requirements or above, then those people generally get the interview, because it's very rare for them to find somebody with a bigger percentage of match. Then if you get an interview, you can again do another informational interview if it's applicable. Then you may have an initial HR screening interview. Then you'll get the online or phone interview, right? These days, a lot of things are done online or phone. And then you'll get if you qualify that, you'll get an in-person interview where you'll have to also do a presentation. And then finally, you get the offer, negotiations, and decision come during that time. So I hope all of you go through all these processes smoothly. And if you have questions, please feel free to reach out. Then here are some similarities and differences that I faced during my postdoctoral and current job interview. So I did do a lot of research on the job, on the lab, on the organizations, and then submitted the application. I got online interview for my postdoc position, phone interview for my industry position. So before that, I again did a lot of research on the people who will interview me. The online interview for postdoc was just the PI. Phone interview was the main hiring manager. Then for for industry, he set me up with all the team members individually for phone interviews. Again, I got to learn a lot about the company, about the role, and what each team member expects from you. So remember, they are expecting things, so you should know what they expect from you. Not everything is clear from just that job description that you can see in the advertisement. So during your in-person interview, they will ask you questions, but you should also ask a lot of questions. So for that, you have to research the interviewers again. Know beforehand who are all going to be in your interview. They usually send you the agenda, so look up those people on LinkedIn and PubMed, and you can also ask them like who will be there and everything. So there will be individual meetings throughout the day, and they will talk about their roles, your roles, expectations, and everything. There will be 1 hour allocated for presentation, and in that presentation there will be everybody. Usually it's the whole organization if it's a small company, or it can be the whole department, the whole lab. Be prepared with good questions. So it's more important to have good questions than knowing all the answers. So some of the things that you need to know are trends, competitors, forecasts. Now, you may not know competitors and forecasts if it's your first job straight out of academia, but you must know the trends because that's the field that you are an expert in. It's okay to say no, and it's okay to say I don't. You don't have to agree to everything they are saying, and you don't have to know everything, but you should show that you're open to learning, you're flexible, you're teachable. They want people who they can train and teach accordingly, because every organization works differently. So no matter how good we were in our previous role, we have to learn new things and conform to the new organization. And then they may discuss terms of the offer, but that doesn't mean that you're getting offered there. That usually doesn't happen, but they may discuss the terms that's going to come if you were to take the role. Then later on, you send acknowledgement and follow-up emails to everybody who spent their time with you. And then finally, you get the offer. Then you can get to negotiate and you can make a decision. So with that, I'll end the presentation and thank you everyone for your attention.
Thank you, Dr. Sen, for talking more about, what it's like to be a field application scientist, especially, how collaborative the role really is, and then also offering the tips and tricks about, applying for the job. So thank you so much. So with that, I will begin the Q&A session, which will be a panel discussion with all of our speakers. So all of our speakers will come back on, and the first question I wanted to start with, so there were a few questions asking about work-life balance. I thought this would be a good question for the entire panel, but specifically, Dr. Bouchier-Hayes, how you kind of manage work-life balance and being, so productive after coming back from maternity leave. So if you want to start off the question, and then we can kind of go around and discuss, how you guys have been managing work-life balance with your current roles.
I will say I'm getting better at it. I think during my PhD and postdoc, there really wasn't any, but yeah, I like, it's almost easier when you have a kid because you're just forced into having to be home at a certain time and all that kind of thing. I think often like researchers who don't have young families, there's an expectation, oh, you have to be in certain amount of hours, and why does work-life balance matter to you as much, which obviously I don't think is correct. But so I think it's just about being able to manage your time as well as you can, and not spending a huge amount of time like doing busy work, like getting into the meat of what you need to do. So one thing that helps me, and I'm in a privileged position where I can do this, is I work from home on Mondays and I use that as my writing day, and I try not to have any meetings that day. And that ever since I started doing that, that really helped me kind of start off the week on a positive note and getting stuff done. And then I could concentrate on all the like admin tasks, answering emails, stuff like that for the rest of the week, and interacting with my lab. But yeah, I think it's just being able to say no to things that aren't going to help you. Putting limits on what you're going to do, like making it kind of obvious. It's like, if you send me an email at 8 o'clock at night, I'm not going to answer it. That kind of thing. And just trying not to let people tell you that you're not. I think the society is getting much better about dealing with this, that you don't have to be there all the time and be seen to be there all the time, but making sure you're just working smart rather than hard. And it's not easy.
Yeah, sometimes it's difficult to set those boundaries for yourself, and kind of negotiate those. So I kind of want to open that up for the rest of the panel in terms of how you've been managing work-life balance. Dr. Potter, do you have anything that you want to add to that?
I think the point about academic flexibility is really important. So my PhD was similar in that I had a timeline for my testing days where I had to stick to those testing days. So if that was on a Saturday or an evening where there was a family event going on, then I had to do the testing first and then maybe try and get to it. But what I've learned, and what I probably wish I had stuck to in my PhD, is that now I know that my managers are happy that if I do have to work in an evening for an open day or for something else, that I can be flexible with my time around that on other days. So I don't need to keep filling that kind of core working hour time if I've been using that time elsewhere. So for the teaching side of things, that's obviously open days, outreach days, other things that happen in the evening. So I think that's a really important part, and making sure, and this is easy to say in hindsight, but making sure you identify a good manager or supervisor that is able to support you with that. So I guess not just rushing in and thinking because they are prestigious academic that means they're also a good manager or supervisor. There's other aspects to that as well that feed into work-life balance.
That's really great insight. Thank you. Dr. Shah, is there anything you wanted to add to that?
I think I want to second both of Lisa and Harry's point, because having good manager, and that's what I always look for my employer, is that does he understand me? I'm a human, I'm not a robo who can work in the lab for 24/7. I have my own life and I want to manage both of them. And at the same time, having a kid and family, it limits you the time wise, because you have daycare times, you have to pick up the kid and drop off, and you have very limited time in between, so you cannot spend 1 hour chit chatting and have a lunch. I just say, hey guys, I need to go, I need to finish this experiment and I need to pick up my kid. So I have to set my boundaries with the time and with the limitation. And if you want to hang out, I'm free in the weekends, but not in the weekdays. I have to manage everything because one day you screw up, the next day kid won't get up on time and you have the whole backlog. So it becomes a robotic life in the weekdays, but I enjoyed it because now I'm bound to the time and I can finish and I can be more productive. Then I'm like, oh yeah, I can do it after 30 minutes. It's okay. Let's have a coffee. Those days are gone.
Yeah. Predictability is not always a bad thing. Dr. Quinn, is there anything you wanted to add?
Yeah, I think I'll echo a few points that people have made, but I just want to say I've been in the horrors of doing, like, I think in the UK, I had a 3-year PhD, so I very much worked 80-hour weeks to get everything done in those 3 years because I had quite a hard stop. But then I moved into a consulting job, and I've seen a few questions in the Q&A about consulting. I think the one biggest take home I took from consulting is that you have to account for every hour that you work, literally to the hour. So they will, even to 30 minutes, sometimes intervals. So you will charge your client that you work with for, 100 hours of work of a researcher to go and, research a project. I think I learned very quickly in 6 months of consulting what it's like to have evenings and what it's like to have weekends back. And I took that through into my postdoc and into industry. And now, for example, we have, sometimes there's a 7 p.m. meeting, 8 p.m. meeting. Generally I ask my boss, do you think, is this an essential one that I attend? Can I catch up with someone after someone else attend? Can they just summarize what happened? And we're really good at that at Eisai. Generally, we only want one person from the group to attend if it's like a late night meeting or something like that. Obviously, if you're presenting, you do need to be there. I'm sure Harry was kind of alluding to that earlier. Like if you're like obliged to attend, you really should. But I think it's kind of carving out your own time and just making sure that you like, I try and get in early, I try and leave early. That's my kind of schedule, but you just kind of work that around everyone else's schedule.
And Dr. Sen, do you have anything you want to add to that?
Yeah, just echo what everybody said. And specifically in my role in the industry right now, because I'm also the territory manager for some of the international territories. So, there's a time difference, and sometimes there are meetings with a customer which cannot be done except for when it's very late night for me. But those, if they are not long-standing, like regularly standing meetings, it's okay if it's a one-time thing. At the same time, my mentors and my supervisors at Active Motif are very mindful of the workload and the work-life balance that not only me, everybody else in the company has. So they are always like checking in and making sure that we are not burnt out. So it's very important to have your supervisors and your colleagues check in on that. I've had very good mentors in academia as well. So there have been cases where you will hear people saying that in academia, people tend to be micromanaging, but in industry, it's more. But it's not like that. I think it depends on the individual person and on the organization. So I have had great mentors in academia too, who were very mindful of all the students' work-life balances. People have had babies, and while they were in PhDs or postdocs, and the PIs and the whole lab was super supportive.
Right. Can I also add to it? I think it is something you have to work out a little bit to separate your work from your home life. The last time I was on one of these kind of work-life balance things, the first thing I was asked was, what are your hobbies? And it just really stressed me out because it's like, I work and I have a child and that's it, hobbies. But if you can find things outside of work, so it's not just all that, whether it's a sport or knitting or whatever, I think it just really helps you kind of have something where you can just not think about work all the time. Because even if you are able to work 9 to 5, sometimes some of us are just thinking about it all the time, and you just I think having whatever you need to kind of take that break away is really good.
Great. Yeah, thank you so much, everyone, for answering that question. This next question is for Dr. Quinn. Do you have any advice on, you mentioned getting in contact with recruiters and being in contact with recruiters. Do you have any advice on how you can do that?
It's a fantastic question. I didn't know how it happened, and then I put open to work on LinkedIn and everything changed. And I think to put, you can put that you're open to work in a certain area, you get job notifications, and actually when you start applying to positions, something happens. I don't know what it is, but I was getting more and more messages that I could even handle when I started that process of actively applying to companies. And then once you find one good recruiter, then they refer you to 10 jobs. So it's really like a snowball effect. So once you start applying, once you sort of set LinkedIn to open to work, and you start reaching out to your contacts, I feel like recruiters can sense it and they are able to find you. So, and then I think in terms of like building relationships with them, I'd often have like a 20-minute call with the recruiters. Some of them didn't want to deal with me because I needed a visa, whereas some were happy with that. So I think I was really upfront and said, this is my situation. I need this visa to work at this company. Is it possible? I would start with that. I wouldn't try and like avoid the fact that I required to getting a visa at some point. So I was really upfront with the recruiters about my situation, and I think they appreciated it. And just being willing to share your resume really quickly with them. And I think Rwik mentioned that earlier around having a short 1 to 2 page resume for industry is super helpful, and having it just, you can have a generic one, but then really make sure it's tailored for each of the jobs. But a recruiter's looking for just a generic resume so they can catch your key skills.
Great, thank you. The next question is for Dr. Potter. So I think there's some confusion with like nomenclature. So you explained your work with pastoral care. Can you explain this a little bit more in detail? Kind of what is it? Is it faith-based, or is it kind of like an academic counselor or advisor?
Yeah, I think that's just the word we use in the UK for an academic advisor, essentially, but it incorporates all of the emotional and physical health that go alongside studying for an academic degree. So it's mental health and well-being, stress management, time management, all of these things that are we're kind of responsible for as academics. And I think the way that it's set up here at UCLan, and probably at lots of other universities as well, is that I, as the year tutor or pastoral support person for that group of students, I essentially act as the central point where students can come into me when they have something that they don't quite understand or an issue that they can't quite solve. And whilst I have been trained on the sort of basics of all of these areas, I'm never going to be a fully trained psychologist. I'm never going to be a fully trained financial advisor, but I can point them towards the right place. So that support of colleagues as well, so the colleagues that are trained on mental health and well-being can then advise me on how I can support the students. So it's all kind of integrated into one big system with the aim of taking in students that are panicking and don't know what to do and getting them to the right place so they can find some sort of solution.
Great. Thank you so much for clarifying and explaining that, because before this I hadn't heard of pastoral care either. So thank you for that. So this is a question for the panel. Maybe we can start with Rwik, but the question is on when you're writing a resume for industry, how do you kind of sell yourself when you don't completely match the skill set that they want? So if you have, for example, a 40% skill match.
That's a great question. So as I said, you have to draw from your transferable skills and really read carefully about what they're asking for, and you'll have to try your best to match. But if it's something like, if it was for a bioinformatics position, then I would definitely not apply because I'm not a bioinformatics expert. But if it's something to do with the bench science and I can see that I can help them in some way, then I would definitely go ahead and apply. You may not know all the techniques, but you can just make a case, and you also have the opportunity to submit a cover letter. So explain there.
Perfect, thank you. Is there anyone else on the panel that kind of wanted to chime in or have any other advice on how you can kind of make yourself an outstanding candidate? Yeah, Dr.
I can throw in real quick. So I applied to an in vivo biology experience job here at Eisai, and I have no in vivo biology experience. So I think I just highlighted that I worked on human samples. So I was like, well, I've done all of these experiments with human. So for me to then do this with mouse, and I explained, like, I don't have any mouse handling experience. So I was very clear with that. And I said, it's not something I'm able to do. Would you still be interested in bringing me on board? And they were. So I think just, having a conversation with the hiring manager, I think they look to hire people with certain skill sets. And, if you're not 100% matched, they can always hire a second candidate for, a different level position. I think they always want to look for someone with 100% of the skill set, but they don't exist. And if they exist, you're not applying to the right level. If you hit 100% of the criteria, you should be applying to a position above that. You should always not meet 100% of the criteria, because otherwise you've got nothing to learn.
That's great advice. Thank you so much. This next question is for Dr. Bouchier-Hayes. So usually you see people are switching from academia to industry these days, but you knew that industry was not for you. So how did you figure out that decision and decide that, industry was not something that you wanted to get involved in?
So I think it was coming from the only lab-based experience I had at the stage when I started my PhD was industry-based. So I just wasn't really exposed to what academic science was like. I also, and I don't agree with this stance, but I was in an atmosphere where it was considered that going to industry was considered selling out. That kind of, I don't know how much that influenced me, but I think just generally it's just my temperament. I like the idea of being independent, being able to kind of work on whatever projects I want to work on. To be honest, with the funding situation, those differences are starting to get a little less diverse. You can only work on things that you have funding for. So I think I had a naive view of what industry research was. It would be like, you'd have a manager saying, oh, you're not getting the right results, move on to something else. In fairness, I've done that to people as well. So, I think it was very early on when I made that switch. So I wasn't really, I think I was showing that to kind of show how I was kind of blindly going through things at that stage. And I had no idea what I wanted to do. There's a lot of pressure on graduate school applicants right now to have a very clear idea of what their career path is. And I think everyone here shows that, you cannot predict what direction you're going to go in.
Great. Thank you. So we have time for one more question, and I thought this would be a nice one to ask the panel and kind of go around. But what is your top tip for interviewing? So let's start with Dr. Shah.
The top tip for the interviewing is just be honest, because I've seen that many people I have interviewed in the current position I am, and they are pretentious, and that's dangerous to the job and to the lab environment. So just be honest. If you don't know, just show the willingness to learn, but don't be oh yeah, I know all these techniques, and then when you're given that technique, you don't know. That's really no. So just be honest and try to show that you're ready to learn your techniques.
Great, thank you. Dr. Sen, let's move on to you.
Sure, I'd echo what Dr. Shah said. You have to be honest and just accept that you don't know. It's okay to say no, I don't know, and be open to learning. And then also be confident of what you are saying. And because when they are hiring you, they are not hiring a student, they are hiring a peer, a colleague. So they should look up to you as someone who they can also trust, and also someone who can, if needed, if the role requires, be on meetings with customers, because you don't have to be a field application scientist only to be meeting with customers. Our R&D scientists also sometimes need to be on the calls. So you have to be that kind of a person who's a subject matter expert and also able to communicate very clearly. So a lot of things I would say, honesty is most important, and then your confidence and just your knowledge, your dependability, your trust and everything.
Great. Thank you. Dr. Potter?
I would say to have some information or some insight into what each of the panel members does. So whether that's one of their top papers, the top paper of your supervisor that you found most interesting. I think academics and scientists are very passionate and love to talk about their own research. So if you're able to show that you're also interested even if that's just in one small part of it, you can show that you've been prepared and you've been proactive in preparing for it.
Thanks. Dr. Quinn?
I've seen a few other questions about the 1 hour seminar, so maybe I'll just focus my answer on that. So the 1 hour seminar, like I had to give, I really focused on the translational aspect of my project. You know, that, yes, I wasn't out there trying to find new therapeutics, but I was out there trying to find biomarkers for Alzheimer's disease. So I really focused on the translational aspect. If you don't have a translational aspect, you need to present a very clearly defined project, start to finish. So that could be, you know, how you helped come up with the idea of a project, how you delivered it, and then how you showed that you completed it. That can be through presenting at a conference, publishing it in a publication, and all of those things. So I think you can use the two. Translational is great, or drug discovery is great, if that's what you're going to be doing in industry, or you can also then do the other angle, which is just a very simple, like, start to finish of a project and how you clearly had a role to play in that. And I think also highlighting any managerial experience. So I highlighted that I'd worked with a few co-ops in the lab. So these were like 6-month undergraduate students, and how I supervise those, because there's a lot of supervision in industry.
Great, thanks. And then we'll end with Dr. Bouchier-Hayes.
So this is kind of more specific to like postdoc positions and the like, but definitely make sure that you get to spend some time with the members in the lab without the PI present. If you're applying somewhere and they're reluctant to let you do that, I would say that's a major red flag for, but it's the best way. If you can go out for coffee, best if you can go out for dinner and, like, you know, everyone's a little loose and, you know, but it's the best way to get to know what the atmosphere of the lab is. Those are the opportunities to maybe kind of get a feel of how supportive the PI is for, you know, work-life balance and all these other things that you may not feel comfortable asking the PI directly. But I think the atmosphere and the mood of the lab is very, very important for how successful you're going to be. Because if you get into a toxic environment, it doesn't matter how much you love the science that you're doing, it's going to be very, very difficult going forward. So that would be my advice. Just try and talk to the people who work there and try and do that independently. If you are doing a virtual interview and you don't, as I said, always try and go to the place, but if everything conspires against you and everything's virtual, set up some individual Zooms with some people who have been there for a long time and ask them questions. Usually people will be honest with you.
Great. Well, thank you. Thank you, everyone, all our speakers, for, you know, your wonderful talks, for sharing not only your journeys, but, you know, the obstacles that you guys went through. A lot of the times we don't hear these. We just kind of see the final piece, and we see these successful people without knowing everything that they've kind of been through. So, you know, we really appreciate you guys taking the time to share your stories and giving your advice and input. To kind of end the ECR event, I just wanted to share a last few slides, kind of just to talk about some resources. So if you liked the content of today's webinar and you want to learn more, Proteintech does have an ECR hub on our website, and that's where you can go to access any past ECR events that we've had. We also offer career development resources, on-demand careers workshops. You can stay up to date with upcoming travel grants and mentorship awards that we offer, and you can also read interviews with past ECRs. I also just want to hand it over to Dr. Sen real quick so that he can talk more about epigenetics and the resources that they offer, active Motif, so that he could talk about the resources that they offer for epigenetics studies.
Right, thank you. So Active Motif is an epigenetics focused company. We've been around for the past 23 years, and we are based in San Diego, actually Carlsbad, California. And we have offices in Europe, China, and Japan, but we have a presence across the globe. And our main portfolio is epigenetics products, epigenetics services, and this Pixel Sonicator. So if you go to our website, you'll find information on all our current products there, and then the services that we offer, where we will do all of these assays for you and provide you with final, fully analyzed bioinformatics results. And then finally the Sonicator. So, and then as I mentioned, we also have a very good resources page where we work closely with academia for blogs, podcasts, and webinars, conferences. So you'll find all of that information there. So please feel free to visit us at activemotif.com. Thank you.
Great, thank you so much, Dr. Sen.
Mm-hmm.
And then lastly, I just wanted to say that as an attendee of today's webinar, you are entitled to receive a free sample of a Proteintech antibody. We will send a link where you can request your free sample in a follow-up email. You will also receive a certificate of attendance as well as the recording of this webinar. So with that, I just want to thank everyone who attended today and all of our speakers. If you have any questions after this webinar is over, feel free to email us at events@ptglab.com. And if you liked today's webinar and want to know more about the events that we do, please follow us on our social media because we do post about our upcoming events there. So with that, I just want to thank everyone once again and hope you all have a wonderful rest of your day.